Next, we have another talk from Rosario Durán. Also her final talk in the course, so you are getting a present too, Rosario. Rosario will leave tomorrow very early in the morning, so today is your last chance to talk to her and discuss your ideas with her if you want.
Hola. Okay, thank you very much again, and hi to everyone. As Herman said, this is my last talk, and I'm leaving this afternoon. So before starting my presentation, I would like to really thank Herman and Nico. They are really great hosts. I had a very nice time here. I hope you are going to organize this in 2 years, because I think it was really great.
Maybe 3 years.
3 years, okay. Okay. I will try not to retire before. So today what I'm going to show you is some of our results. This is our research. And what I tried to do here is to make like a tour for around different interactomic approach we have been using in our lab for a lot, let's say in the last 10 years or so. And some of them are very simple, or others are a little bit more complex. But I try to show you for each of these interactomic approach the advantages and the controls, and to take some idea of what they were good for in our hands. So our interest is based on this suborder of organisms that is called Corynebacterineae, which is a little bit difficult to pronounce. But in fact, it's a suborder of bacteria that have very important particularities that I'm going to show you. And we started with this work many years ago, and with the results of each interactome, we start moving from studying a single protein kinase to study processes like cell division and elongation. So, which is the particularity of this group of bacteria? Okay, the first thing is the cell wall. This bacteria has a very thick and impermeable cell wall that is composed by many layers not found in other bacteria. And in particular, these mycolic acids that are very long fatty acid, very hydrophobic ones, are a characteristic of this group of bacteria that include many important human pathogens as Mycobacterium, tuberculosis, and Corynebacterium diphtheriae. So this is one of the things that is very distinctive of this group of bacteria. And the other one, or one I would like to focus on, is the phosphorylation systems. As you know, any cell has phosphorylates proteins in order to regulate many processes. And in bacteria, protein phosphorylation, or the classical bacterial protein phosphorylation systems, involve two residues, histidine, and aspartic acid, and these two proteins, the histidine kinases and the response regulators. So this was done by us long time ago, and what you can see is that for different bacteria, the number of genes that codify these histidine kinases and response regulators is variable, can be as high as 70, but some others have slower numbers. But if we compare this with this gray bar, that is the genome size, you can see that in this group here, there is really, this phosphorylation system are underrepresented. There is comparatively low number of these kinases and regulators. Oh, I touched the wrong button as well. Okay. And then in this group of bacteria, at the same time, the number of serine-threonine kinases that phosphorylates serines and threonines, and sometimes also tyrosines, it's at the same time overrepresented. So one of the first questions we would like to answer is, which is the role of these serine-threonine kinases? And focusing on Mycobacterium tuberculosis, because it's a very important pathogen, we know that this pathogen has 11 genes codifying serine-threonine protein kinases, and we have focused our attention in one of these kinases, named protein kinase G, because it was very early demonstrated that this protein has an important role in virulence, and it was proposed to be a virulence factor. And we have studied this protein, and we have found it's a complex protein, has several domains. It has a kinase domain here that is able to phosphorylate other proteins, mainly by transferring the phosphate group to serine and threonine residues. But it is also able to autophosphorylate its own sequence. And in this, we found that in this N-terminal, there were up to 4 threonine residues that were phosphorylated by the kinase, and that are a docking site. They are a site that can be recognized by other proteins in the signaling cascade. So, the correct one? So we try to find substrates of this kinase. This is just a brief introduction to give some biological context to the interactomes I'm going to show. So we first perform experiment with radioactive ATP to find proteins that were able to be phosphorylated by this kinase, and we identified this substrate that is called GERA, that is a small protein that has this domain here, that is FHA domain, that specifically recognizes phosphothreonine in other proteins. So we have demonstrated using different biochemical techniques the interaction and the phosphorylation, the mechanism of the interaction and the phosphorylation of GARA by PIC and G. So this is the kinase, this is the N-terminal that is autophosphorylated, and as I told you before, these phosphate residues act as a docking site for the FHA domain of the substrate that recognize phosphorylated threonines, and then place the phosphorylation site of the substrate facing the active site that then is phosphorylated, and after phosphorylation is closed. It becomes in a closed conformation where the phosphorylation site is recognized by their own FHA domain, and this switch off all the interactions of this protein. So the first thing that we wonder here is, okay, this is an FHA protein, recognized phosphothreonines, which are the proteins that are recognized by GARA in Mycobacterium tuberculosis. So to answer this question, we made the easiest experiment that can be done. We just produce GARA as a recombinant protein with an E-stack, and we immobilize this recombinant protein and use this to capture downstream partners of GARA. So it was a very simple experiment. I'm not saying that this always worked, but we were very lucky. We have to have luck, and we were. So this was even visible in a one-dimensional shell. So here is the control that has no recombinant protein. This is the one that has GARA, and these two lines are, because we performed this experiment in two strains. One is Mycobacterium tuberculosis, and the other is the model organism Mycobacterium smegmatis. And as you can see, we have GARA here and not in the control. And there are several bands that are only present when you put GARA. This does not mean that these are interactors at all, because many times using a blank column is not a good blank. Sometimes you unmobilize an unrelated protein, and you also can have several bands. But in this case, these were strong bands. One of these was PIC and G. So this is was very important at that moment for us, because it's a positive control. So we know that PIC and G interacts with GARA, so we start relying more in the rest of the interactors, and then we just cut these bands, identify the proteins, and these turn out to be two enzymes that forms part of the central metabolism of the bacteria. So we then, of course, validate this. We made assays to show that this is a direct interaction using surface plasmon resonance and recombinant proteins. And we made enzymatic assays to see which is the role of GARA in these two proteins. In fact, there are three there. So, and we found that this small substrate can inhibit two key enzymes that regulate the entrance of glutamates into the Krebs cycle and activates another one that goes in the other side. So it was acting synergically on three enzymes that regulate this, the balance between nitrogen and carbon metabolism in the bacteria. And then we said, what happens is GARA is phosphorylated. Okay, when GARA is phosphorylated, we don't have any interaction at all, and all this, all the regulation of these activities is blocked. So I show you this very old experiment because I think that there's an important message here, because sometimes you can do a very simple experiment and have very good results and reach very good conclusions. In fact, these experiments and many others that came afterwards let us...
Thank you, Rosario, for this amazing story in your life in Interactomics.
Something like that.
Questions for Rosario? We're going to have the first break of the day, and we return here at 11:15, Nico. Is that okay? Yes.